Previous class we completed up to protein synthesis, phytohormones, everything. Today we complete from biotechnology, genetic engineering to mushroom cultivation. So we discussed some bits regarding this one. First one, a glycoprotein produced by a virus in the infected host cells. A glycoprotein produced by virus in the infected host cells is interferon. What is interferon? A chemical substance produced by virus in the host cells, infected host cells, to prevent the infection of other viruses is interferon. So a glycoprotein produced by a virus in the infected host cells is not probe, it is not antibiotic, it is not hormone, it is interferon, a chemical secreted by virus infected cell. Virus itself in the infected cell to stop the infection of other viruses is interferon. This is a matching tabular form. Match the following enzyme and use: peptide, rennet, proteases, monosodium glutamate, flavoring agent, leather softener, meat tenderizer, cheese preparation and artificial sweetener. A3, peptide, it is meat tenderizer. Peptide is widely used in meat tenderizing. Rennet is used in making cheese. In the cheese production, rennet is widely used. Proteases are popularly used as leather softeners. Monosodium glutamate is a flavoring agent. So peptide, meat tenderizer, rennet, cheese preparation, proteases, leather softener, and monosodium glutamate, flavoring agent. Which of the following are nutritionally influenced diseases? Nutritionally influenced diseases means the diseases caused due to the deficiency of various nutrient substances. Whenever food material get deficient or various nutrient material get deficient, these type of diseases occur. They are diabetes, hypertension, cancer. So diabetes, hypertension, cancer are nutritionally influenced diseases. AIDS is caused due to some other reasons. So diabetes, hypertension and cancer are nutritionally influenced diseases. Proteomics, genomics are supported by. Proteomics means the science that deals with the study of knowing the chain or sequence of amino acids in a computer is proteomics. Genomics means the concept or science of studying genomes by using computer technique. Genome is a haploid set of chromosomes. So proteomics is chain or advance or what you call sequence of amino acids in a protein by computer is proteomics. Genomics is the sequence of genes with the help of computer is genomics. It is not gene therapy, it is not bioremedy, it is not biopesticides, it is bioinformatics. So bioinformatics is the concept which supports proteomics and genomics. How many genes are introduced in type A variety of rice for production of vitamin rich A, golden rice variety? So to produce golden rice variety, how many genes of vitamin A are introduced into type A variety means 4 genes. How many genes are introduced into type A for the production of vitamin A rich golden rice variety is 4 genes are introduced to produce golden rice variety. Mushrooms can be cultivated on domestic waste, industrial waste, agricultural waste, hospital waste. So mushrooms can be cultivated on any waste recycling. Mushrooms and SCP are very much useful. So domestic waste, industrial waste, agricultural waste, hospital waste, all can be used in the growing mushrooms. Roots of biotechnology are present in. Roots of biotechnology means how the genes are transferred and other things. It is not PCR technology, it is not fermentation technology, it is not chromatography technology, it is rDNA technology. rDNA technology means a gene is isolated, it is inserted into a vector. Vector is allowed to multiply continuously to produce same copy of genes which are called clones. Clones are introduced into a host, they are allowed to multiply within the host, along with the host. Transformed cells are formed. Like this method is conducted. So roots of biotechnology are present in rDNA technology, first option. The gene cloning can also carried out in a computerized machine called thermocycler by a method called. The gene cloning, multiplication of genes after inserting the bit of DNA into a plasmid or some vector producing the same copy of genes is called gene cloning. The gene cloning can also be carried out in a computerized machine called thermocycler. Thermocycler is a machine which is widely used in gene cloning by a very popular method called polymerase chain reaction. It is also called PCR, polymerase chain reaction. Carboxylic cycle, citric acid cycle, polymerase cyclic reaction are wrong. Polymerase chain reaction is a method in which a thermocycler is used in the artificial gene cloning. One of the following techniques is used to separate DNA fragments. DNA fragments are cut by restriction endonucleases, either in the form of what you call even cut or in the form of staggered cut by producing blunt end or fleshy ends or palindromic sequence. All the bits are grouped together, not by hybridoma technology, not by gene cloning, not by molecular forming, but by gel electrophoresis. So, one of the following technique is used to separate DNA fragments and group them into a particular place is called as gel electrophoresis. The method which is used to select a desirable gene in the group is called Southern blotting. When a cell synthesizes new proteins after intake of foreign DNA, such a cell is called. When a cell starts synthesizing new proteins after the intake of foreign DNA, whenever a bit of DNA is inserted into a cell, that cell where the bit of DNA is inserted and incorporated into that particular DNA is called as transformed cell. So, when a cell starts synthesizing new proteins after the intake of a foreign DNA, such a cell is not called cloned cell, mutated cell, modified cell, but it is called transformed cell. Genes which confer antibiotic resistance on bacteria are located in. Genes which contain antibiotic resistance on bacteria are located not in chromosomal DNA, not in RNA, not in polysome, but plasmid. Plasmid is a vector with special antibiotic resistant genes by which they are identified during selection of transformed cells. Arrange the usage of the following in a sequence during isolation of gene. First, restriction endonuclease. First, phenol treatment is given. Whenever phenol treatment is given, the DNA bit comes out. After DNA bit coming out, with the help of restriction endonuclease, EcoR1 or some other enzyme, the DNA bit is cut. So, first is always 3, adding phenol treatment. First, enzymatic degradation of cell wall, then cell membrane is disintegrated by using detergent lysis, then phenol treatment is given, DNA comes out, then DNA is purified by a method called gradient centrifugation, then restriction endonuclease is used to cut the DNA into bits. So, first phenol treatment, then restriction endonucleases, then all the bits of DNA are grouped together by gel electrophoresis, finally Southern blotting. Desirable bit is selected by Southern blotting. So, first phenol treatment, then restriction endonuclease, then gel electrophoresis, and finally Southern blotting. Arrange the usage of following chemical techniques in a sequence during genetic engineering. Genetic engineering, detergent lysis, colony hybridization, gradient centrifugation, calcium chloride. So, first all these are used: detergent, gradient centrifugation, colony hybridization, calcium chloride. First will be calcium chloride, then first detergent lysis 1, then usage of gradient centrifugation, then colony hybridization, and finally, so 1, 2, 4, 3 will be the answer. First and third will be the answer for this. Arrange the techniques in sequence following isolation of genes: electrophoresis, gradient centrifugation, detergent treatment, phenolic treatment. First detergent treatment, then phenolic treatment, detergent treatment to what you called disturb the plasma membrane, then phenolic treatment to take out the DNA from the cell, then gradient centrifugation to clean the DNA, and finally gel electrophoresis to group the DNA. So, first detergent treatment, then phenolic treatment, then gradient centrifugation, then electrophoresis. 3, 4, 2, 1, third option. These are biopesticides. Biopesticides are nuclear polyhedrosis virus. There is another one, chloropolyhedrosis virus, nuclear polyhedrosis virus. Bacillus megatherium is a bacterium which is used in curing tobacco leaves. Curing tobacco leaves is done by megatherium, though it is not biopesticide. Bacillus thuringiensis, which is inserted into cotton to form BT cotton, bit cotton. So, biopesticides here are NPV, nuclear polyhedrosis virus, and Bacillus thuringiensis 1 and 3. The technique of used for isolation of gene are techniques used for isolation of gene in genetic engineering are colony hybridization is nothing but selection of transformed cells. This is the last step which is done with the probes and without probes. So colony hybridization is selection of transformed cells as the last step in genetic engineering. Gel electrophoresis, southern blotting are techniques used for isolation of gene. Gel electrophoresis is after cutting the DNA into bits either as palindromes or what you call blunt ends or flesh ends, all are grouped together by gel electrophoresis. Desirable bit for the process is selected by the most important method called southern blotting because it was explained by a scientist called Southern. This is not used as vector in rDNA technology. This is not used as a vector in rDNA technology. Thermocycler is not used as a vector. Thermocycler is an instrument where polymerase chain reaction is conducted, that is artificial cloning of gene is done. DNA is isolated, attached to its special plasmid. Plasmid is allowed to perform multiplication. This is done artificially, which is called polymerase chain reaction. Whereas phos and cosmid are widely used as vectors in rDNA technology. So, which is not used in vector in rDNA technology is thermocycler. Phos and cosmid are used. So, answer is 1. Thermocycler is not used as a vector in rDNA technology. Plasmids are isolated by using, first, a cell is how the plasmid must contain 4 properties. It must have a low molecular weight. It must have some resistance to antibiotics. It must possess a single specific restriction site for the activity of restriction enzyme. And fourth property is it must show a true replication in any host where it is inserted or sent. How the plasmid is taken out means first bacterial cell is treated with lysozyme. With lysozyme cell wall is broken. Then a chemical called EDTA, ethylene diamine tetraacetic acid. After that, the cell is centrifuged in sodium lauryl sulfate. By all these things, the plasmid will come out. So, here plasmids are isolated by using ethylene diamine tetraacetic acid, EDTA and lysozyme. What is the use of calcium chloride here? Answer is 1 and 3, but what is the use of calcium chloride is after the formation of recombinant DNA, it must be sent into a bacterial cell which is known as transformation, and bacterial cell is called transformed cell. It is sent into the bacterial cell. Cell wall is not permeable for rDNA. So, cell wall is treated with calcium chloride. Then cell wall becomes permeable and rDNA is easily sent into the bacterial cell. So, plasmids are isolated by using ethylene diamine tetraacetic acid and lysozyme 1 and 3. A probe in genetic engineering. The transformed cells or colony hybridization is done by 2 methods. Without probes and with probes. Without probes means all the transformed cells are grown in a medium without antibiotics. Then the cells start showing antibiotic resistant genes. Then when they are transferred to a medium with antibiotic, immediately the resistant cells will survive and all the other cells will disintegrate. That is without probes. With probes means single stranded DNA or single stranded RNA is radioactively labeled and sent into the medium. They start searching for their complementary base sequence. By that, a cell can be easily identified. So, a probe in genetic engineering is single stranded DNA and single stranded RNA, not a double stranded DNA. So, answer is single stranded DNA, single stranded RNA. Example or answer is 1 and 2. This is an assertion reasoning. The sequence of nitrogen bases in the single-stranded sticky ends of DNA double helix formed by the activity of E. coli is adenine, adenine, thymine, thymine. So, the sequence of nitrogen bases in the single-stranded sticky ends of DNA double helix formed by the activity of EcoR1 is AATT. This enzyme cuts the DNA strand between G and A sides of the palindromic sequence. So, because it cuts the DNA in between G and A, AATT are obtained. So, A and R are correct. R is the correct explanation of A. This is another assertion reasoning. Plasmids are the most commonly used vectors in genetic engineering. In genetic engineering, plasmids are the most common vector that are widely used. Plasmids are easily isolated and introduced into the host cell. Because they are easily isolated from the bacterial cell, and after that they are easily introduced into the host cell by using simple chemicals like EDTA, lysosomes, sodium lauryl sulfate and calcium chloride. Plasmids are the most commonly used vectors in genetic engineering is correct. Plasmids are easily isolated and introduced into the host cell is correct. So both A and R are correct. R is the correct explanation of A. Sodium lauryl sulfate is used in isolation of plasmid. Definitely sodium lauryl sulfate is widely used in isolation of plasmid because the centrifugation of bacterial cell done in the presence of sodium lauryl sulfate only. It denatures the plasmid DNA is wrong. It does not show any effect on plasmid DNA. So A is true, R is false. Sodium lauryl sulfate is used in the isolation of plasmid is correct. It denatures the plasmid DNA is wrong. So A is correct, R is false. Lambda phases are used as cloning vectors. In genetic engineering, lambda phases are widely used as cloning vectors. Most of these phases DNA is not required for the formation of phase particle in the host cell. So a part of DNA is not at all useful in the production of phase cells. So if that part is removed and a bit of DNA, foreign DNA is attached there, nothing will happen. That's why lambda phases are widely used in cloning vector. So lambda phases are used as cloning vectors. Most of the phase DNA is not required for the formation of a phase particle in the host cell is correct. So A and R are correct. R is the correct explanation of A. EDTA is used to isolate the donor gene. EDTA is used to isolate the donor gene. It helps in the dissolution of bacterial cell wall that is used to carry out transformation. EDTA is not used to isolate the donor gene. It is used to isolate the gene from the cell. So EDTA is used to isolate donor gene is wrong. It helps in the dissolution of bacterial cell wall and used to carry out transformation is correct. So A is false, R is true. cDNA is used as probe in genetic engineering. cDNA is complementary DNA which is formed from DNA or which is formed from messenger RNA by reverse transcription. Whenever DNA produces mRNA, it is called transcription. Whenever mRNA again produces DNA, it is called reverse transcription. By reverse transcription, complementary DNA is formed. So complementary DNA are widely used as probes in genetic engineering. The sequence of complementary DNA is complementary to the sequence of desirable gene. Because the sequence of complementary DNA is complementary to the sequence of desired gene, it is widely used as a probe in genetic engineering. So A and R are correct. R is the correct explanation of A. A probe is always single-stranded DNA. DNA used as a probe must be labeled. So a probe is always single-stranded DNA is wrong. A probe may be a single-stranded RNA also, but DNA is used as probe must be labeled. Without labeling, we cannot identify the correct complementary sequence in the transformed cells. So a probe is always single-stranded DNA is false. DNA used as a probe must be labeled is correct. So A is false, R is true. DNA fragments generated by restriction enzyme can be separated using the following technique. After using the restriction endonuclease enzymes, DNA is cut into small bits or fragments. After that, they are separated and grouped together into one point. It is not gas chromatography, it is not liquid chromatography, it is not centrifugation, it is gel electrophoresis, second option. Which of the following enzymes joins two pieces of DNA that have been cut by the same restriction endonuclease? Same EcoRI cuts the DNA in between GA site in between G and A. Not only in the bacterial cell but also in the, not only in the plasmid of bacterial cell but also in the vector. And both are reconnected at the same point because of identical sticky ends by an enzyme called ligase. That's why this method is called DNA ligation. So which of the following enzymes joins two pieces of DNA that have been cut by the same restriction endonuclease is not kinase, polymerase, lyase, it is ligase. The process where a section of DNA is inserted into a plasmid or bacteriophage and then replicated to numerous copies of insert is known as. Whenever a gene is isolated and sent into a plasmid or vector anywhere, if it starts producing same copy of genes, that method is not called splicing or spooling or transformation, it is cloning, popularly called gene cloning. Recombinant DNA technology is used for transfer of foreign genes between unrelated organisms is correct. Transfer of foreign genes. A gene from human being, insulin producing gene, can be taken out, inserted into Escherichia coli, and that Escherichia coli is allowed to multiply repeatedly to produce the same human insulin, popularly called humulin. So transfer of foreign genes between unrelated organisms. The best example is production of human insulin, humulin, by Escherichia coli. Transfer of prokaryotic genes into a eukaryotic cell. It is also done by recombinant DNA technology. Development of new strains within a very less span of time. New strains can be produced. All of the above. So recombinant DNA technology is used for all of the above. Molecular forming. By the transfer of genes, special plants are produced called transgenic plants. These transgenic plants are useful in various methods. Producing cultivated plants using radioactive isotopes is wrong. Artificial synthesis of DNA and RNA is wrong. Cloning of bacteria is wrong. The correct answer of molecular forming is commercial production of specialized medicines, chemicals, and antibodies using transgenic plants. Producing cultivated plants using radioactive isotopes is not molecular forming. Artificial synthesis of DNA and RNA is not molecular forming. Cloning of bacteria is not molecular forming. It is commercial production of specialized medicines, chemicals, and antibodies using transgenic plants is called as molecular forming. Colony hybridization is a method useful for. Colony hybridization is a method useful not for producing recombinant DNA. Recombinant DNA is DNA ligation. Producing a progeny by conjugation between two different bacteria is not correct. Producing new mutant varieties is not correct. Colony hybridization is a method useful for the detection of the presence of a gene in a transformed cell. So whenever it is done without probes or within a medium containing antibiotics or by using single-stranded DNA and RNA, the detection of presence of a gene in a transformed cell is known only by a special method called colony hybridization. So colony hybridization is a method useful for the detection of presence of a gene in a transformed cell. First option. A single-stranded RNA or DNA segment which is radioactively labeled and used to locate complementary DNA sequence from an organism is called. A single-stranded DNA or RNA segment which is radioactively labeled and used to locate complementary DNA as a sequence from an organism is called not genomic DNA library. What is genomic DNA library? Transformed cells are kept in a particular place, that is called genomic DNA library. Plasmid is extra chromosomal genetic material present in a bacterial cell. Cosmid is not. So answer for this is probe. Crossing over that occur during meiosis similar to the laboratory technique of gene manipulation called genetic engineering in. Crossing over that occur during meiosis, that is in Pachitin, is similar to the laboratory technique of gene manipulation called genetic engineering in appearance of gene recombinations. Crossing over always cause recombinations. Resulting in the appearance of always undesirable combination of genes is wrong. Resulting in the appearance of only desirable combination of genes is wrong. In lacking endonuclease activity during the process is wrong. Appearance of gene recombinations is correct. First option. During the isolation of desired genes in the process of recombinant DNA technology. During the isolation of a desired gene in the process of recombinant DNA technology, purified DNA is precipitated out ultimately after the addition of chilled ethanol. This purified DNA represents. So during the isolation of a desired genes in the process of recombinant DNA technology. During the isolation of desired gene in the process of recombinant DNA technology, purified DNA is precipitated out ultimately after the addition of chilled ethanol. This purified DNA represents nucleotide strands plus intranucleosomal histone proteins only wrong. Nucleotide strands, all histone proteins is wrong. Only nitrogen base is wrong. Only polynucleotide strands without histone proteins is correct. So, fourth option is correct. It is an assertion reasoning question. The protruding ends generated by staggered cut. The protruding ends generated by staggered cut are also called sticky or cohesive ends, and this kind of cutting is obtained by a limited number of restriction endonucleases like E. coli. The protruding ends generated by staggered cut are also called sticky or cohesive ends, and this kind of cutting is obtained by a limited number of restriction endonucleases like E. coli is wrong. So many restriction endonucleases are discovered. They cut the DNA at a particular place in between two nitrogen bases and resulting in the formation of sticky ends. Staggered cut yields fragments in which one strand of DNA double helix extends some bases beyond the other cut end. So, definitely by staggered cut, so suppose DNA double helix with so many nitrogen bases, when staggered cut is done, the DNA shows this type. AATT, TTAA. This is called palindrome sequence or identical sticky ends. The single stranded termini of one strand shows nitrogen bases. The single stranded termini of one strand shows nitrogen bases which are exactly equal to the nitrogen bases of other strand in a reverse direction is called palindromic sequence. 4 to 6 net nitrogen base pairs of one strand are exactly equal to the nitrogen bases of other strand in a reverse sequence is called as palindromes. So, the protruding ends generated by staggered cuts are also called sticky or cohesive ends, and this kind of cutting is obtained by a limited number of restriction endonucleases like E. coli is wrong. Staggered cut yields fragments in which one strand of DNA double helix extends some bases beyond the other at cut end is correct. So, A is false, R is true. PCR technique carried in thermocycler is comparable to this natural process. What is PCR techniques? Multiplication of gene. Multiplication of gene is nothing but multiplication of DNA. The small bit of DNA which was attached to plasmid, and plasmid allows to multiply to produce same number of copies of genes, which is called gene cloning. So, PCR technique carried out in thermocycler, polymerase chain reaction technique carried out in thermocycler is comparable to this natural process, not crossing over of meiosis, not cell cycle, not RNA technology or DNA technology, but S phase. S phase is the most important phase in interphase where DNA get doubles. So, here also doubling of DNA is taking place. So, polymerase chain reaction techniques carried out in thermocycler is comparable to this natural process, means fourth one, S phase. A linear DNA molecule having both non-sticky ends has 4 5' AAGCTT 3' palindromic sites. If this DNA molecule is cleaved with a specific restriction endonuclease between A and A of 5' AAG 3' sequence, how many hydrogen bonds may be removed in total? 5' to 3', how many? First time when AATT are cut, 4 are removed. So, here 5 into 4, 20, 20 plus 20. So, in total 40 hydrogen bonds are removed when a linear DNA molecule having both non-sticky ends has 4 5' AAGCTT 3' palindromic sites. If this DNA molecule is cleaved with a specific restriction endonuclease between A and A of 5' to AAG to 3', the sequence how many hydrogen bonds may be removed in total means 40 hydrogen bonds are removed. EcoR1 specifically recognizes ZAA site in DNA molecule, and it can be cut in the same way in this way. Between G and A in 3' to 5' direction is wrong. Between A and A in between 5' to 3' direction is wrong. Between A and G in 3' to 5' direction is wrong. Between G and A in 5' to 3' direction is correct. So, EcoR1 specifically recognizes ZAA sites in DNA molecule, and it can cut the same in this way between G and A in 5' to 3' direction. Fourth option. Enzymes used in the isolation of purified DNA. Cellulases, pectinases, high powered detergents, phenols, proteases, ribonucleases, chilled ethanol, restriction endonucleases, DNA ligases. So, these many enzymes are given, and we must identify what are the enzymes used in the isolation of purified DNA. Answer is A, D and E. What is A? Cellulases and pectinases, which breaks the cell wall. Proteases, D, and E, ribonucleases. These are used in the isolation of purified DNA. Cellulases and pectinases, proteases and ribonucleases. Now, those are some of the bits in the case of what you call DNA technology, genetic engineering, and 4 steps in genetic engineering: isolation of gene, insertion of gene into a vector, introduction of rDNA into a special host, and isolation and selection and screening of transformed cells, then transgenic plants, some of the important biotechnological concepts and other things. Coming to tissue culture and other aspects: autoclave, sodium hypochlorite, laminar airflow chamber, soil right, biofertilizer, inoculation chamber, coconut shells with organic matter, steam sterilizer, disinfectant. A4, autoclave. Autoclave is a steam sterilizer. Autoclave is the component in which the nutrient media or anything is kept and heated at 121 degrees centigrade, 15 lbs pressure for 15 to 20 minutes. The total nutrient medium becomes microorganisms free, that means sterilized medium. So, A4, autoclave, steam sterilizer. B, sodium hypochlorite. Sodium hypochlorite is a disinfectant. Whenever you take an explant, any plant material taken out from the plant is called explant. This explant will be definitely having so many microorganisms on its surface because it is growing in open climatic condition. So, it is cleaned or disinfected by special chemical called sodium hypochlorite. Then, laminar airflow chamber. Laminar airflow chamber is an inoculation chamber where nutrient medium is kept and sterilized explant is taken and kept on the medium by removing the cotton plugs. So, that is laminar airflow chamber. Soil right. Soil right is coconut shells with organic matter. A powder of coconut shells with organic matter is called soil right. Why? Because whenever the plants are produced after tissue culture, they are grown in a place because they do not have any cuticle as they grow in 100% moisture. They are grown in pots with soil right, coconut shell and organic matter covered by polythene bags. After some time they develop cuticle and grown in gardens. So, A4, B5, C2, D3. Cellular totipotency means ability of a cell to give rise to a tissue is wrong. Ability of an organism to reproduce its young ones is wrong. Ability of a cell to develop into an organism by regeneration is wrong. Retaining the cell division power by a cell. The capacity of a cell to produce a complete plant is cellular totipotency. Totipotency, potentiality of a cell to produce a complete plant is cellular totipotency. So, retaining of cell division power by a cell to produce numerous cells in an organism is called as cellular totipotency. Cellular totipotency was first experimentally demonstrated by F.C. Stewart in. Cellular totipotency was first experimentally demonstrated by F.C. Stewart, not in secondary phloem of carrot root, not in secondary xylem of carrot root, not in primary parenchyma of carrot root, but in the secondary phloem of carrot stem. In the secondary phloem of carrot stem, cellular totipotency was first experimentally demonstrated by F.C. Stewart. Aseptic seedling can be developed from a seed which are germinated on. Aseptic seedlings can be developed from seeds when they are germinated on not basal medium, medium with auxins only, medium with cytokinins only, medium with fruit juices. Medium with auxins only, aseptic seedlings are developed from the seeds. During the production of aseptic seedlings, the seeds are surface sterilized with. During the production of aseptic seedlings, the seeds are surface sterilized with, not with sodium hypochlorite, because sodium hypochlorite is a disinfectant which cleans the explant obtained from the plant. Not potassium metabisulfate, not copper oxychloride. It is 0.1% mercury chloride. During the production of aseptic seedlings, the seeds are surface sterilized with 0.1% mercuric chloride. Sodium chloride hypochlorite is used in. Sodium hypochlorite is used in not sterilizing the medium. It is used in sterilizing the explant. So, answer is not 1, it is 2. Not inoculation, not morphogenesis, sterilizing the explants. Rhizogenesis is due to. Rhizogenesis means formation of root system. Whenever in a medium auxins are more, root system is formed, cytokinins are more, shoot system is formed. So, whenever a callus, an undifferentiated mass of cells is treated with these hormones, when auxins are more, root system is produced, rhizogenesis occurs. When cytokinins are more, shoot system is produced. So, whenever auxins are more and cytokinins are less, rhizogenesis occurs. Whenever cytokinins are more and auxins are less, collagenesis occurs. So, rhizogenesis is due to more auxins than cytokinins in a medium, not Zibberlin's nor more cytokinin than auxin. The answer will be collagenesis, formation of shoot system. Abscisic acid is not at all useful in any tissue culture method. So, rhizogenesis is due to more auxins than cytokinins. Which of the following cannot be obtained on a basal medium? Which of the following cannot be obtained on a basal medium? Complete plant from callus is impossible. Germination of seed cannot occur. Development of seedling from seeds, but it is callus from explant. Callus from explant. Third one. Soil right is used in the following step of tissue culture. Just now we told what is soil right. Coconut shell with many organic components is called soil right. It is used in the following step of tissue culture, not in inoculation, not in incubation, not in sterilization, but acclimatization of plantlets. Small plants produced during tissue culture are called plantlets, and these plantlets are absolutely without cuticle because they grow in 100% moisture. Cuticle development is done by keeping them in small pots covered by polythene bags. Plants are covered by polythene bags and they are grown in soil right. So, acclimatization of plantlets. Soil right is used in the following step of tissue culture, acclimatization of plantlets. Fourth option. Nutrient medium is solidified by adding. Whenever a nutrient medium is formed in the form of liquid medium, the liquid nutrient medium is converted into semi-solid medium. Semi-solid medium, not by adding naphthalene acetic acid, not by adding sodium hypochlorite, not by keeping explant. It is only by adding agar-agar. It is a special chemical polysaccharide obtained from rhodophysian algae Gelidium and Gracilaria. Those two rhodophysian algae gives rise to agar-agar, which helps in the solidification of nutrient medium. So, nutrient medium is solidified by adding not naphthalene acetic acid, not sodium hypochlorite, not explant, but by agar-agar. Third one. Popular nutrient medium which is used in tissue culture. Very popular nutrient medium which is used in tissue culture is MS medium. MS medium is Murasige and Skoog medium. MS medium is Murasige and Skoog medium, the most widely used medium in tissue culture techniques is MS medium, Murasige and Skoog medium. MS medium, GS medium, Arnon's medium, Sachs medium. It is MS medium, Murasige and Skoog medium. Any part of the plant or any part of the plant which is introduced to culture medium to grow into full-fledged plant or organ in vitro. In vitro means within the laboratories. So, any part of the plant which is introduced to culture medium to grow into full-fledged plant or organs in vitro is called not a propagule, not a clone, not a plantlet, it is popularly called explant. So, any part of the plant which is removed from the plant and used in tissue culture experiments is popularly called as explant. Fourth option. Synthetic seed does not contain zygotic embryo, embryoid, endosperm, nutrient medium, perisperm and gel or sodium alginate layer. The synthetic seeds, what is a synthetic seed or artificial seed? By tissue culture method, somatic embryoids are produced. The embryos produced from somatic cells are called somatic embryoids. The somatic embryoids are encapsulated by sodium alginate. So, any somatic embryoid encapsulated or covered with sodium alginate component is called synthetic seed or artificial seed. So, synthetic seeds does not contain A. Zygotic embryo, C. Endosperm, E. Perisperm. So, zygotic embryo, endosperm, perisperm are completely absent in synthetic seeds or artificial seeds. From a virus infected vegetatively propagated plant, virus free plants can be produced by. From a virus infected vegetatively propagated plant, virus free plants can be produced by. Virus free plants can be produced by not by embryo culture or by anther culture or by organ culture, it is by shoot tip culture. Shoot tip culture is widely used to produce virus free plants from virus infected plants. Mismatch of the following. Shoot tip culture production of virus free plants is correct. Synthetic seeds or alginated embryoids is correct. Somaclonal variations means variations due to gene recombinations, none. Here the answer is 2. Variations not due to gene recombination, variations due to somatic cell development. So, shoot tip culture production of virus free plants is correct. Synthetic seeds or alginated embryoids is correct, none is given here, and somaclonal variations are variations which are obtained during tissue culture methods by vegetative cell implement or what you call growing vegetative cells in a culture medium. Variations due to gene recombinations are not somaclonal variations. So, mismatch in the following is second one. Somaclonal variations due to gene recombination. This is a matching column. In the matching column, agar-agar, sodium hypochlorite, sodium alginate, soil right. Soil with organic matter, solidification and liquid medium, coconut shells with organic material, disinfectant used for surface sterilization of plant, encapsulation of somatic embryos. Answer is 2. A2, agar-agar, solidification of liquid medium. Whenever a liquid medium is added with agar-agar, immediately the liquid medium converts into semi-solid medium. Sodium hypochlorite 4, a disinfectant used for surface sterilization of explant. A disinfectant used for surface sterilization of explant is sodium hypochlorite. Sodium alginate 5, encapsulation of somatic embryos. So, during tissue culture, embryo-like structures are produced called somatic embryoids. When these somatic embryoids are encapsulated with, that means covered with a special chemical called sodium alginate, it results in the formation of synthetic seeds or artificial seeds. Synthetic seeds or artificial seeds can be stored very effectively. They can be transported very effectively, and above all, they are made to germinate without any much resting or dormancy period. Soil right. 3. Coconut shells mixed up with organic matter. So, agar agar, solidification of liquid medium. Sodium hypochlorite, disinfected used for surface sterilization of explant. Sodium alginate, encapsulation of somatic embryos. Soil right. Coconut shells with organic material. Through this method, more number of female plants can be produced in papaya. Papaya is a dioecious plant. Carica papaya is a dioecious plant. In a dioecious plant, separate male and female plants are present. When the plant comes to flowering stage only, we know which one is male plant and which one is female plant. Vegetatively, not much differences will be there. In Carica papaya, which is a female plant, which is a dioecious plant, female plants give fruits. One male plant can be enough to pollinate many female plants. So, many female plants are produced within a short span of time by tissue culture. So, through which method? More number of female plants can be produced in papaya, not by spraying ethifon, not by genetic engineering, not by polyploid breeding, it is by tissue culture. Virus-free plants can be obtained through shoot tip culture. Virus-free plants can be obtained through shoot tip culture. Virus-free plants obtained through shoot tip culture are resistant to viruses. Virus-free plants can be obtained through shoot tip culture, and those plants which are obtained through shoot tip culture are resistant to viruses. So, both A and R are correct. R is the correct explanation of A. Plantlets produced through tissue culture should be acclimatized to prevent from desiccation. Plantlets produced through tissue culture must be acclimatized to prevent desiccation. Why? Because they do not have any cuticle, because they grow in 100% moisture. Suppose they are directly introduced to the environment because of heat and other things, the total plant will undergo desiccation. Total water is lost and plant will die. So, A is correct. Plantlets develop under high percentage of moisture content during tissue culture. Plantlets develop under high percentage of moisture content during tissue culture is wrong. So, A is true, R is false.